I'd like to talk about high-throughput sequencing to provide a background for everything else we're going to be talking about the rest of the day. We're going to start with a little overview of high-throughput sequencing technologies and some of the terms we'll be using. Then we'll go on talking about the collection strategies for high-throughput sequencing, how you should design your experiments to make the best use of them. Then we'll talk about the data formats throughout the process, and then how you would go about processing that data. First, how does high-throughput sequencing work? What we'd really like to do is just take a piece of DNA, stretch it out, read the whole thing all at once, but for a variety of reasons, that's just not practical. The current state of the art is to split it up into many short reads of a few hundred base pairs instead of looking at millions of base pairs at once. We take these small fragments and we clone them. We make many copies, and it is the result of this cloning that is what is given to the sequencers. Then the sequencers give us the sequencers read these short reads and give us short sequences of letters A, C, G, and T. Now, today's sequencing machines have improved dramatically over the decades. We have so much data now. So we need to think of sophisticated ways to process those. Some of the terminology. So we have these DNA fragments that I mentioned earlier. When we have all these clones, we call them inserts. We run them through some processes in order to make them suitable for the sequencers. At this step, they're called libraries. We may have many libraries per sample. Then we load these libraries onto the different lanes of a flow cell, typically add the library to multiple lanes. And the reason we do this is so we have a lot of redundancy. If something goes wrong in the process, we have some sort of backup to work with. The sequencer gives us these short reads. The sequencer has no idea where these short reads are located on the genome. It just knows what the letters are. We need to figure out some way to align them, some place to map them, put them in the correct location on the genome. There are lots of sophisticated algorithms and programs that do this, and we'll talk about this later in our best practices. All right, here's an example of a short region of the genome with some aligned reads. Let's start at the bottom. This is our reference genome. It's displayed in 4 colors just to make it more obvious which bases it's talking about. And then above it, all those gray bars are individual reads that come from the sequencer. These have all been aligned. These have all been located in the correct locations. The gray color means that it is the same as the reference. When you see those T's in the middle, if it's a different color, it means that it is found that those bases are different from the reference. In this case, it's pretty clearly mostly reference everywhere, except for those T's in the middle. And for that column, at that particular genomic location, about half of them are T's, and half of them match the reference, which we see as a C below. So it's a pretty clear CT heterozygote. All right, how should we make the most of our sequencer? Now, it'd be really nice if we could just do deep sequencing of whole genomes of all of our samples, but we don't have infinite time, we don't have infinite money, other people want to use the sequencers. We need to figure out what can we do within our budgets. For the same amount of time, money, and sequencer use, you could have these 2 different basic strategies. You could do a single sample very deeply, or multiple samples shallowly. We have the same number of total reads here, and depending on our analysis, we may choose one or the other. The basic trade-off here is that if you sequence a single sample deeply, you will find out a lot about that particular sample, but you will not know anything about your population in general. With multiple samples, you will get more variance on your population in total, but the individual calls for the individual samples may be a little less accurate because you don't have enough evidence for those specifically. Going into some more detail on the single sample sequencing. This is the ideal. If you have your big budget, you can do your deep coverage of a whole genome. Here you have the ability to discover 99% of the variation in the genome. Your probability of discovering common alleles and singletons is both over 99%. But the problem is that you have large storage costs. You're going to be, for each genome you have, you're going to have to store 100 billion sequence bases. This will give you great sensitivity for your heterozygotes, your homozygotes, but maybe this is too much of a cost. What's another alternative? You could do a low-pass whole genome sequencing design. This requires much less use of the sequencer, but it provides for certain power. For common alleles, you're still going to do pretty well, but for the singletons, you're going to be doing a lot worse. If you only have about 4 reads for every particular location, you're going to miss a lot of things, particularly heterozygotes, because you don't have enough evidence to say this is confidently half one, half the other, although homozygotes are pretty good. If, on the other hand, you want to do an exome sequence instead, you want to focus on the 1% of the genome that biologists tell me is pretty much the most interesting, you can do this for a lot cheaper. The target here is to get about 80% of the exome at 20x coverage, and this will allow you to make a lot of discoveries. You're going to have pretty good power for common alleles and for singletons. You're only looking at a very small part of the genome, but it's generally the most interesting part. And you're going to have a lot less storage cost here. All right, let's talk about the different data formats that are involved in high-throughput sequencing. All right, here's the basic process. You have your sequencer that gives you base calls. It tells you, which bases it sees from the reads. It applies quality scores, how confident it is in each of those reads, and that's a FASTQ file. From there, you perform alignment or assembly to the genome reference, and then you have mapping information and also mapping qualities, and those are your SAM and your BAM file. SAM stands for sequence alignment mapping, and BAM is the binary format, which is good because it's much smaller, it's compressed, it's indexed, you can access it a lot quicker. From there, you do variant calling, statistical analysis of determining which are the actual variants, and then you have the variant call format file. This is a text file by default, but it can also be compressed and indexed for speed of use. All right, detail of the FASTQ format. It is first we have an identifier. Each read has its own unique identifier. Then we have the actual bases the sequencer has read. And finally, the qualities. What is the confidence that the sequencer has in its reads? This is a Phred scaled quality score, which I'll talk about in a moment. All right, so what is Phred scaling? It's basically a nicer way to be able to talk about confidence, about error rates. We have some very high quality sequencers now, where you might have an error rate of say 0.001, 0.00001, but that's just going to be cumbersome to be talking about all day. So we have this transform into a quality score. So if you're 90% confident that it has made a correct call, we call that quality 10. If you're 99% confident, that's quality 20. So we're talking about quality 40 versus quality 50. That's a lot friendlier. So that encoding in the FASTQ is not really for human readability, but it's very easy for a computer to process that. We see the Phred scores, yes. So as the accuracy of the sequencer's confidence goes up, we have a higher Phred score. That's the blue line, and of course the error rate that it believes it has is the opposite. After FASTQ, we have the aligned version of the reads. That's the BAM. This is a very nice format because it's the industry standard. It's vendor neutral. Everybody writes the same format. So no matter who does the sequencing, everybody can all read these files in the same way. So a BAM record starts with a read name, which is the same as from the FASTQ. But then it also has a locus. We see that this particular read has been mapped to chromosome 1 at that location. And it also provides some alignment information. That's called a cigar string. That describes how it aligns. Here we have 23 matching bases, a gap of 6, and 10 matching bases. Then after that, we have the actual read, and the quality score directly from the FASTQ. And finally, we have some metadata applied from the aligner. The band format also has a header. This is very important because you're always aligning to a particular genome reference. So the beginning tells you about which reference was used, all the different contigs in that reference. After that, we have read groups. What this is a description of which sequencer was used, which lane, which sample, which library. This is important in case we have, something went wrong during processing a particular lane, a particular machine. Later, trying to recalibrate based on error rates, this will be very important information. Each read is associated with a specific read group. The variant call format is a text file. It has a header. So what it describes mostly is various annotations. The info fields are annotations at the site level. They're specific calculations it has done at the particular genomic location. The format fields are per sample. Finally, in the last header line, the one that has CROM over there, that has the default column headers for the rest of the file, and it ends with the sample names. Here we have 3 samples, NA1, 2, and 3. The VCF entries below it, I'll talk about the first one a little bit. So we see that is mapped to chromosome 20 at position 14370. It has that particular ID sequence. Because it is some known variant, it has looked up in a database, probably dbSNP in this case. We have the alleles, G, the reference allele, and the alternate allele, which has been seen by at least one sample in the variant call. We have examples of different kinds of variant calls here. The first one is a G to A SNP. Second one is an A to G or T multi-allelic SNP. The third one, we just see the T for the reference and a dot. What that means is that none of the samples in this analysis differ from the reference. And that last one is a complex indel, where we have both an insertion and a deletion in this sample set. All right, and how do we process these data? How do we process these data formats? So, seems like a relatively straightforward process. We have a sequencer that reads and gives quality for the reads. We have aligners that align and give qualities for the alignment. Based on all that information, we can just make, it should be pretty easy at that point to figure out what is an actual variant and what is nonsense. Well, you get situations like this. This is an actual IGV screenshot. There's lots of reads here, all covering the same location, but they're all telling us different things. What does this mean? I cannot tell you what this means. Is this just noise? Are these actual variants? Maybe there's some of both. So how do we solve that? Well, that's what the GATK is for. Basically, we designed this toolkit in order to solve problems like these, to make these things tractable. Hopefully, it will be useful to you. Now Geraldine will come back to talk to you about how to get started using the GATK. Thank you.